Welcome to the video on flagella. So flagella is another accessory structure, meaning not all bacteria have it. Those that do, they have these long whip-like structures that allow them to move through their environment. Flagella are built primarily from the protein called flagellin, and they include three main parts. The filament, which extends away from the cell, like here that you see the whip-like structure, a hook that connects the filament to the cell surface, and then the basal body, which anchors the flagellum and acts like a motor. So you can't see those last two here. As the basal body or the motor rotates, it causes the filament to spin and propel the bacteria forward. So I like to think of it like a boat propeller. They spin, and when they're having a counterclockwise rotation of the flagella, the bacteria projects forward. Okay, and so we call this running. But now imagine a boat that's going just straight in the water. Now imagine trying to turn that flagella just suddenly, or I'm sorry, that motor, the opposite direction, right? It's going to stutter because it's going smoothly one direction. And if you just flip the switch to have it go the other way, it's going to be some rough movement, okay? And that process is called tumbling when it's kind of changing direction and orienting itself. So we say that the bacteria movement follows a pattern called running and tumbling, which is what you see here in this picture. So again, during a run, the flagella rotate in this coordinated way that bacteria moves in a relatively straight direction. But then during a tumble, when the flagella moves a different way, the movement becomes less coordinated. So the bacteria kind of stops and randomly reorients itself and then will eventually start running again. So by alternating between runs and tumbles, bacteria can respond to environmental signals through chemotaxis. So again, it's following a chemical attractant or even a repellent, but we tend to talk more about it moving toward favorable conditions, such as nutrients, or like I said, it can move away from more harmful substances. So flagella can be arranged in different patterns around the bacterial cell. So we call this a flagellar arrangement. And because it's an accessory structure, I want to start with the terminology for no flagella at all. So if you look at this bold word, okay, atrichus, A means without, and then trichus, I believe, is Latin for hair. So think of it like without hair, because remember, they're these long whip-like or hair-like structures. So a few other arrangements. So monotrichous is going to be just one of these hairs or one flagella. Amphi, think of like ambidextrous. Amphi means there's some on both sides. So in this case, there's just one at each end. And then lophotrichous means there's a tuft of flagella at only one end. And notice with these three, the flagella are located at the polar ends of the bacterium. And then peritrichous, I remember this one because the flagella is just covering all over the bacteria, so it looks hairy. So I think of it as hairy peri. And so it's important to know these arrangements because as we go throughout the course, we're going to be talking about how we classify bacteria based on different things. And one way we can classify or describe them is based on their flagellar arrangement. And um, either having flagella or not is going to dictate, whether the bacteria can move around medium or not. And then, like our peritrichous flagella tend to be very modal because they have just so much flagella that they just move really, really quick. And so we are going to talk a little bit more about these in lab when we're looking at growth patterns and certain media that we can essentially stab the bacteria into it with an inoculating needle. And you'll notice that some bacteria that don't have flagella, you're only going to see growth in that stab line because they can't go anywhere else. Whereas others are going to be able to move away from that stab line because they have structures like flagella. All right, y'all, that's it for this video. Let me know if you have any questions.